Hello friends, welcome to my channel Pharmagyan. Friends, in this channel you can learn about pharmaceutical guidelines, pharmaceutical industrial practices, processes, documentation practices, and many more about pharmaceutical industries. Friends, please don't forget to like, subscribe and share my channel Pharmagyan, and press bell icon given in bottom right corner to get latest updates directly in your notification panel when I upload new videos. So next we will discuss about the video.
Part 8 of 8. Sampling methods. In order to demonstrate that the plant equipment is verified clean and meets the predefined acceptance criteria, sampling and analysis should be carried out using the methods described in the following sections. Justification should be provided for the selection of the appropriate verification technique on a case-by-case basis. A combination of the two methods is generally the most desirable. For all methods, the sampling points should be fixed in a manner such that the true contamination of the equipment will be reflected. Swab sampling, direct surface sampling. Swab sampling of the direct surface is designed to test small sections of the equipment surface for the presence of residues. Samples should be taken from all main equipment items, and since swab sampling does not cover the entire equipment surface area, justification should be provided for the choice of the area for swabbing. Typically, a small area of the cleaned equipment is swabbed with a material according to a predefined method, that is swab material, solvent and technique. The swab sample can then be extracted and examined using a suitable analytical method. The quantified residue obtained from the sample is then extrapolated to the whole equipment. See equation 6. Sampling methods, continued. It is important that the validation of the swab sampling is performed on the same surface, material, Polish grad.d.d, area in DM2, and with the same materials as the routine sampling of the equipment. That the choice of swabbing material considers extractable materials that could interfere with the expected residue. To ensure that the sampling points represent the worst-case areas of the equipment. The disadvantage of this sampling method for often complex API equipment is that difficult-to-reach areas, for example sealings, condensers, transfer pipework, may not be accessible by swabbing. Nevertheless, these areas may be the critical areas for the determination of the amount of residue in the equipment. Equation 6. Sampling methods, continued. M amount of residue in the cleaned equipment in mg. WF recovery rate for the whole chain swab, analytical method, for example 0.8 for 80%. Dot the entire inner surface of the equipment in DM2. Me amount of residue, for example previous product, in the sample I in mg. C gross amount of residue in the sample I in mg. CBI blank of the sample I in mg. To establish the blank, a swab, or several swabs, can be treated in the similar way as a sampling swab except swabbing of the contaminated surface. Usually one and the same blank can be used for all N sampling swabs. Area swabbed by the swab I in DM2. N number of swab samples. I sample identifier, current number from 1 to N. The first production batch of the following product may be sampled and analyzed for impurities, for preceding product, since chromatographic analytical methods will typically be used, for example HPLC, GEC, TLC. Sampling methods, continued. Rinse or wash solvent sampling. In cases where swabbing is not possible, for example restricted access, swabbing may be substituted by the analysis of final rinse solutions. Rinse samples can be used to determine the carryover of residues over a large surface area and cover all main process items including transfer pipework. In cases where swab sampling is not practical, it is acceptable to analyze only rinse samples, however this should be justified as part of the validation study. This section outlines the quantitation of the amount of residue remaining in the equipment after cleaning based on the amount of residue in the last rinse of the routinely used cleaning procedure. The residue amount in the equipment can be assumed to be equal to the amount of residue in the last wash or rinse solvent portion. The assumption is based on the worst case consideration that a further rinse, or any reaction, would not remove more than the same amount of residue present in the analyzed rinse sample. Sampling methods, continued. The advantage of the rinse sampling method is the whole equipment will be reached by the solvent, including difficult-to-reach locations that cannot be disassembled. Therefore, if appropriately designed, this method will give the best indication of the amount of residue remaining in the equipment. For quantitation, a solvent sample, for example 1 liter, is removed and the residue in the sample is determined by a suitable analytical method, which can then be extrapolated to the whole equipment according to equation 5. Equation 5, M equals V times C, CB, where M amount of residue in the cleaned equipment in mg, V volume of the last rinse or wash solvent portion in liters, C concentration of impurities in the sample in mg, L, CB blank of the cleaning or rinsing solvent in mg, L. If several samples are taken during one run, one and the same blank can be used for all samples provided the same solvent lot was used for the whole run. Sampling methods, continued. Stamps. In this exceptionally used sampling method, coins, or stamps, are placed on appropriate sampling points in the equipment during the manufacture of the previous product and during cleaning. After cleaning, the contamination on the coins can be analyzed and the overall contamination can be calculated by extrapolation to the whole equipment. For quantitation, the coins may be firstly swabbed followed by further analysis of the samples. Analytical methods. A sample isolated by either of the sampling methods discussed in section 8.3 should be analyzed by a suitable analytical method, for example HPLC, GEC, GEC-MERS, TLC, dry residue, TOC, UV, titration, conductivity or pH. The suitability of the method can be documented by appropriate validation as detailed in section 8.2. A combination of analytical methods can be used if appropriate. For example, evaporation of the solvent sample and analysis of the dry residue by another method, for example HPLC, can enhance the sensitivity of the final analytical method by a factor 106. Alternatively, the use of several methods, for example titration, HPLC, can provide the required specificity. Thanks for watching the videos. End of the APIC Cleaning Validation Guidelines. Note: this guidance document is transcribed from APIC Active Pharmaceutical Ingredients Committee website. This video is only for educational purpose, not for any professional use. Thanks.